What if the most sophisticated predator in your local ecosystem doesn't have teeth, legs, or eyes? What if it just sits there, perfectly still, and lets chemistry do the work? There are plants out there that produce toxins, mimic pheromones, and build physical traps refined over millions of years of evolutionary pressure. And most people walk right past them without a second thought. We're looking at 10 of them today, because once you understand how these systems work, you'll never look at a flowering plant the same way again. Number 1. Venus Flytrap. Most people picture the Venus flytrap as a simple spring-loaded snap trap, like something you'd find in a cartoon. Touch it once, and it closes. That's not what happens. Each lobe has trigger hairs inside, and a single touch does nothing at all. 2 touches within about 20 seconds start the closure, but the trap won't commit to full digestion until those hairs get triggered 5 or more times, because that confirms something living is actually inside and worth the energy cost. What the plant is counting are electrical pulses, action potentials, the same type of signal neurons fire in animal brains. If prey stops moving, digestion pauses. If it keeps struggling, enzyme production ramps up. Number 2. Pitcher plant. The pitcher plant doesn't move at all. It just builds a structure and makes that structure progressively harder to escape from, which is a completely different approach. Sarracenia flava lines its rim with nectar laced with coneine, the same narcotic alkaloid found in poison hemlock. Insects feed on it, lose motor control, and slide down into a tube where the walls turn waxy and angle downward so there's nothing to grip. The digestive fluid at the bottom contains a wetting agent that destroys surface tension, so even an insect that lands on the pool sinks immediately. Nepenthes raja from Borneo scales this architecture up to pitchers that hold over 3 liters of fluid, with confirmed prey including rats, frogs, and small birds. Number 3. Sundew. Drosera, the sundew genus, looks like it's covered in water droplets. That's the point. Insects see the glistening tentacles and read them as moisture, which is a resource worth landing on. Those droplets are mucilage, a sticky gel that won't dry out in heat or rain, and every time an insect struggles against it, more tentacles bend inward to add contact points. A fly that lands on Drosera rotundifolia can be fully enveloped within 30 minutes as the leaf curls around it. The mucilage also carries digestive enzymes and compounds that suppress the insect's immune response, so digestion starts before the prey stops moving. Over 200 confirmed Drosera species grow on every continent except Antarctica, which tells you this strategy works across a wide range of climates and prey types. Number 4. Corpse flower. Amorphophallus titanum doesn't eat anything. It needs carrion beetles and flesh flies to carry its pollen, and to get them moving in the right direction, it runs one of the most precise chemical deceptions we know of in the plant world. The flower heats itself to near human body temperature and releases a blend of sulfur compounds: dimethyl trisulfide, dimethyl disulfide, indole, and trimethylamine, each one targeting a different smell receptor in its target insects. The heat isn't incidental. The plant burns stored carbohydrates specifically to push those odor compounds further through the air. Insects arrive expecting a carcass. They find pollen instead, and the whole window runs less than 48 hours before the flower shuts down completely, and the mating instincts of bees turn out to be an even more reliable system to exploit. Number 5. Hammer orchid. Male thinnid wasps in Australia spend much of their adult lives tracking female pheromone trails. Dracaea glyptodon, the hammer orchid, has essentially copied that signal and broadcast it as its own. The flower produces a chemical blend that closely matches female thinnid wasp sex pheromones, and the labellum, the lower petal, is shaped and textured to feel like a female wasp's body. A male wasp locks onto the scent, attempts to mate with the flower, and pollen masses called pollinia attach to his body. He flies off, finds another hammer orchid, tries again, and delivers the pollen. Each hammer orchid species targets one specific wasp species, so the signal isn't a broad attractant. It's a narrow, precise chemical key cut for a single lock. Number 6. Bee orchid. Ophrys apifera looks like a bumblebee landed on a flower and just stayed there. That visual resemblance is enough to pull male euchra bees in for a pseudocopulation attempt. And when they press against the labellum, they pick up pollen and carry it to the next flower they try with. In Northern Europe, though, those bee species don't exist, so the plant just handles it internally. The pollinia bend forward on their own and deposit pollen directly onto the stigma without any insect involved. So it's running a full deception system in the south and a self-sufficient backup in the north simultaneously, which tells you how much pressure shaped that deception in the first place. Number 7. Rafflesia. Rafflesia arnoldi has no leaves, no stem, no roots, and no chlorophyll. It lives entirely inside the tissue of a host vine, pulling water and nitrogen directly from its vascular system, and the only part of it you'd ever see above ground is the flower itself, which can stretch over a meter across. Carrion flies arrive expecting a carcass or an egg-laying site, crawl through the interior, and pick up pollen. But successful pollination needs 2 flowers of opposite sex blooming within fly travel distance during the same narrow window, and that doesn't happen often. So Rafflesia dumps enormous quantities of pollen when it does bloom, betting on volume to compensate for timing that's almost never in its favor. Number 8. Dead horse arum. Helicodiceros muscivorus runs a 2-phase system that most deceptive plants don't bother with. On day 1, it heats its spadix to near mammalian body temperature and releases a blend of oligosulfides that smell like rotting carrion. Blowflies arrive, crawl inside the spathe looking for a carcass or somewhere to lay eggs, and downward-pointing hairs block them from leaving. While they're trapped overnight, pollen from the male flowers coats their bodies. By morning, the plant cools, the smell fades, and the hairs relax so the flies can leave. What researchers found is that the heat itself attracts blowflies, not just the smell, because warm surfaces read as fresh kills to their sensory systems. Number 9. Doll's eye. White baneberry. Actaea pachypoda looks like something out of a fairy tale. Bright white berries, each one marked with a single black dot, sitting on thick red stalks against green foliage. The name doll's eye fits perfectly, and so does the danger. Every part of this plant carries cardiogenic toxins that disrupt cardiac muscle, and for most mammals, including us, the berries can cause cardiac arrest at sufficient doses. But here's what's actually going on. The plant isn't trying to kill everything, it's filtering. Birds resistant to the toxins eat the berries and pass seeds intact. Mammals eat them and get poisoned, which trains local populations to avoid them over time. The bright display isn't an invitation, it's a test. Number 10. Passion flower. Passiflora doesn't lure anything and doesn't trap anything. It manages behavior across multiple species at once, which is a different kind of manipulation entirely. Heliconius butterflies lay eggs on passion flower leaves because their caterpillars eat the foliage. The plant grows small yellow structures on its leaves that look nearly identical to Heliconius eggs. And female butterflies won't lay on a leaf that already looks occupied because their own caterpillars are cannibalistic. No toxin needed. At the same time, extrafloral nectaries on the leaf surface secrete sugar that recruits ants, which then patrol and attack caterpillars. The plant pays for its own security force. And Anisosceles aloepes, a bug that feeds on passion flower vines, waves its bright legs at predators using chemical defenses it likely picked up from the plant itself. Every plant on this list faced the same basic problem. Not enough nutrients, not enough pollinators, or too much pressure from herbivores. None of them solved it with a brain or a nervous system. They solved it with chemistry, structure, and timing refined across millions of years of pressure. What the data shows is that being rooted in one place doesn't make a plant passive. It makes deception more necessary. A Venus flytrap counting electrical pulses and a passion flower faking butterfly eggs are running the same core calculation. How do I get what I need without giving anything real back?